Researchers identified the hormone that controls glucose levels in blood related to appetite and weight. The new hormone acts on the brain, waking up the hunger in the hypothalamus to control appetite and body weight. The study, led by Dr. Atul Chopra, the medical geneticist at Baylor College of Medicine, discovered the new hormone called asparagine. This hormone opens possibilities for treatment in overweight people where there is no effective treatment exist till now. Neonatal progeroid syndrome. We discovered asparagine when studying individuals affected by a rare medical condition called neonatal progeroid syndrome, said Chopra. Corresponding author, Caroline Wieslaw, scholar and assistant professor of molecular and human genetics and molecular and cellular biology at Baylor College of Medicine. We found that patients with neonatal progeroid syndrome have a mutation in the FGN1 gene that causes them to lack a small piece of the fibrin-1 protein. In individuals without the FGN1 mutation, this small piece, which we named asparagine, is cut and released into the circulation from the end of the protein. The fundamental features behind neonatal progeroid syndrome is thin or very low body weight. To understand the cause, Chopra and his colleagues analyzed the food intake patterns and metabolic rate of the patients. We evaluated the patient's energy balance in a chamber called an indirect calorimeter, said Chopra, who also is a member of the Dan L. Duncan Comprehensive Cancer Center at Baylor. This allowed us to measure how much food they ate relative to the number of calories they burned every day. This test gave us a clear answer. Compared with individuals with normal weight, neonatal progeroid syndrome patients have abnormally low appetite. Because these patients have low blood asparagine levels due to their mutations, we wondered whether asparagine was in fact necessary to maintain normal appetite in people. Functionality of asparagine. The researchers investigated and genetically engineered mice to carry the same genetic mutation the patients have. The result was mice showed the human condition as low blood asparagine levels, low appetite, and were very thin. In this mouse model, we were able to reverse the low appetite by administering asparagine to the mice, Chopra said. The researchers turned to colleagues at the USDA/R's Children's Nutrition Research Center who specialize in studying brain circuits that control appetite. In collaboration with Dr. Yang Su's laboratory, we found that in the brain, asparagine interacts with neurons in the appetite center of the hypothalamus, Chopra said. Two types of neurons involved in appetite control. One type, the AGRAMP neurons, stimulates appetite while the other type, the MC neurons, suppresses it. Asparagine works on both types of neurons in an opposite manner. It activates appetite stimulating AGRAMP neurons and it deactivates appetite suppressing the MC neurons. However, the effects of asparagine on AGRAMP and the MC neurons appear to be quite unique, as we did not find asparagine changing the firing activities of other appetite-regulating neurons, said Su, Associate Professor of Pediatrics and Nutrition and of Molecular and Cellular Biology at Baylor and corresponding author of this work. The resulting effect of these two asparagine actions in the brain is an increase in appetite, a phenomenon that is deficient in individuals and mice with neonatal progeroid syndrome, although we know some intracellular signals like CAMP required to mediate asparagine's appetite-stimulating effects on AGRAMP neurons. The receptors for the hormone remain to be identified, Su said. In addition to studying the neonatal progeroid syndrome who have low levels of asparagine, also studied individuals with obesity and found that they had increased levels of blood asparagine. For more news, visit spinanews.com.